Your blood type is determined by the presence or absence of specific surface antigens on red blood cells. An antigen, often a protein, is something that will provoke an immune response. And so our red blood cells have antigens, A antigen or B antigen, that determine our blood type. There are quite a few antigens on our red blood cell, but in terms of blood type, we really only talk about three: the A antigen, the B antigen, and the Rh antigen. Rh stands for rhesus monkey because it was discovered in the rhesus monkey, but often we call that the D antigen. And remember, an antigen is just a protein that's going to provoke an immune system response. So we call our blood type the ABO blood group, and it's based on the presence or absence of the antigen. So there are four ABO blood types: type A, type B, type AB, and type O. Well, if you have type A blood, your body will produce, your plasma has antibodies that will attack the foreign antigen, which would be type B. If you have type B blood, you have the surface antigen B, but you will have anti-A antibodies in your plasma. If you have type AB blood, you have no antibodies. And if you have type O blood, you have antibodies against A and against B. Agglutination is clumping. And this occurs when these antigens are exposed to the antibodies. So here in this example, we have a red blood cell with the antigen B. And if it is exposed to the anti-B antibodies, these antibodies, which are these Y-shaped proteins, will stick to the red blood cell and to that B antigen and cause them to clump. So we cannot give type A blood to someone who is type B because type B blood contains A antibodies. This is called a cross reaction, and it will cause damage throughout the body, clumping throughout the body, and it will destroy tissue and cause death. The Rh blood group is based on the presence or absence of the Rh or D antigen. If you are Rh positive, you have the antigen. If you are Rh negative, you lack the antigen. So you have to, for your full blood type, you have to include your ABO blood group as well as negative or positive. So blood distributions throughout the United States and the world differ. You can see that of Black Americans, Caucasian Americans, Chinese Americans, blood group O is most common. And, AB is least common. And it looks like most people are Rh positive. So it's easy to determine your blood group. In lab, we did it with the Eldon cards, where you mix a drop of your blood with solutions containing antibodies. And you can visually see the clumping or agglutination that occurs. So if you had type A blood, it would clump with anti-A antibodies. With O blood, you saw no clumping or agglutination at all. And this blood typing is necessary to avoid transfusion reactions, which are cross reactions that occur from transfusing mismatched blood. So donor and recipient blood types must be compatible. That is, they will not cross-react or clump. So let's look down here at the AB positive blood. When mixed with the A antibody, there's clumping. Mixed with B antibody, there's clumping. And mixed with the RH antibody, there's clumping. This person with AB positive is the universal recipient, meaning they can receive blood from all blood types. On the other hand, O negative blood is the universal donor. They can donate blood to all blood types. So this ABO blood system and the RH positive and negative antibody, RH positive and negative system is genetically determined. If you're RH positive, you don't automatically have anti-RH. An RH negative person will not automatically have antibodies against RH positive the way we have with the ABO blood system. But if you're RH negative and you're exposed to RH positive red blood cells, then you will develop anti-RH antibodies. What would happen if a patient with type AB positive blood were given type A positive blood? There would be no reaction. What types of antibodies are found in the plasma of a person with O positive blood? Anti-A and anti-B. So there's a bit of a problem with this RH factor for moms and newborns. Hemolytic disease of the newborn is a red blood cell related disorder caused by a cross reaction between fetal and maternal blood types. So our blood type is determined by combining the genes from both mom and dad. So a child can have different blood type from either parent. It's kind of easy to show genetically how that happens, but we don't get into the genetics of it here. During a pregnancy, the mother's antibodies can cross the placenta and attack the fetal red blood cells. This is a condition called hemolytic disease of the newborn, hemolytic blood breakdown disease. And this is a problem that usually involves an RH negative mother who has carried an RH positive fetus. So if an RH negative mom mates with an RH positive man, the fetus can be RH positive. It's not a problem with the first pregnancy because those fetal cells are not mixing with maternal blood whatsoever. But during birth, bleeding at the placenta or the uterus mixes the fetal and maternal blood. The mother is exposed to the baby's RH positive blood. And because she's exposed, she will produce anti-RH antibodies. Approximately 20% of RH negative moms who carry RH positive kids are sensitized within 6 months of delivery. But again, that's after the first child has been born, so it does not affect the first child. But if she has another pregnancy with an RH positive fetus, her antibodies can cross the placenta. The placenta is an organ that forms between the fetus and the mom. It's kind of like the middleman, and nutrients and gases and wastes pass through the placenta from the mom going to the fetus. So the antibodies can pass, cross the placenta, and destroy fetal red blood cells, causing anemia. As the fetus grows, there's more and more need for red blood cells. And so erythroblasts, which are precursors to the the red blood cell, enter the bloodstream before they're mature, and this leads to erythroblastosis fetalis. And there is a high fatality rate with this. So in order to prevent this, the mom can be given a RhoGAM shot during pregnancy and after delivery. And that will destroy her antibodies against the Rh factor. So again, this is only a problem with an Rh negative mom who has an Rh positive fetus. Which of these disorders is caused by Rh factor incompatibility? Hemolytic disease of newborn. So we want to look at the different types of white blood cells. These are leukocytes. There are 5 types. Never let monkeys eat bananas. Neutrophils, lymphocytes, monocytes, eosinophils, and basophils. These are complete cells. They have nuclei and other organelles, but they have no hemoglobin. They do not function to carry oxygen. So white blood cells circulate for only a portion of their lifespan. Sometimes they lurk in organs, and sometimes they circulate. They can get in and out of the bloodstream through this process called diapedesis. They kind of squeeze out between the capillary cells. The capillary is made up of simple squamous epithelium, a single layer of flat cells, and the white blood cells just kind of squeeze out between those cells. The white blood cells are attracted to chemicals. So say a chemical that's produced by an infection, the white blood cell will migrate toward that infection because of positive chemotaxis. And 3 of the types of white blood cells are capable of phagocytosis. They engulf and destroy toxins, cells, debris. The eosinophils are not real abundant in the blood. Neutrophils and monocytes are. A macrophage is actually a monocyte that has escaped the bloodstream through diapedesis and entered the surrounding tissues. So again, white blood cells only stay in circulation a short period of time. They're mostly located in connective tissue where infections often occur. They can squeeze out of the bloodstream through diapedesis. They show positive chemotaxis, so they will move towards damaged tissue or infection. And neutrophils, eosinophils, and monocytes are capable of phagocytosis. White blood cell count on average is 7,000 per microliter, and this will increase in response to infections or allergies. And a differential will determine the number of each white blood cell count in a sample, and so it would be reported as a percentage. So the technician would count 100 white blood cells and determine what type each white blood cell is. There are 2 classes of white blood cells, granular and agranular. The granular leukocytes look granular because they have these cytoplasmic vesicles that absorb stain, and the agranular ones do not absorb the stain. The granular leukocytes are neutrophils, eosinophils, and basophils. Monocytes and lymphocytes are agranular. The most abundant leukocyte is the neutrophil. 50 to 70% of leukocytes are neutrophils.